All right, I think it's time to start, and I hope everyone is doing well. So I'm so excited to be a part of Expo 2024. It's my third time to attend in this international conference. And before getting started, I would like to thank organizer of this amazing informative conference, and they are bringing researcher teacher from different parts of the world together. And I'm very happy to be a part of that. So the topic that I selected this year is about the computational neuroscience and how mathematical language or differential equation is gonna be used to model communication between the neurons. So I'm gonna start by just some a few introduction about anatomy of a neuron. So in the left-hand side of this slide, you can see a neuron cell. And I start from the one of the most important part, which is called cell body or soma, which is the central part of the neuron that contains nucleus, as you see here. This part is a red part, and it's responsible for basic life process of the cell. And then we can see some branches around this uh, cell body. These are called dendrites. So dendrites are a branch, and these are some branches which are protruding from the cell body. And actually, they are receiving the all of these signals, messages from other neurons or any receptors, and what they do that they are transmitting all of these signals to the cell body and then to the other part of the cell. And we have axon, another part of the cell, which is this long and slender projection, which is coming from cell body. It actually conducts all of these electrical messages that coming from cell body to the other part of the cell, which is, we are going to talk about them here after that. We are going to talk about now axon terminals, these are actually small branches. As you see, the size is smaller than dendrites. And these are at the end of the axon. And what they do, actually, they release neurotransmitter to transmit signals to the other cell. And we have this synapses here. These are like junction, which are like connecting two neurons to each other. And these junctions or synapses are the site where information are transforming from one neuron cell to the other one. And at the end, we are talking about neurotransmitters, which are chemical messengers that transmit signals across the synapses. And they release this vesicle in the axon terminals, and they bind to the receptors on the receiving neurons to transforming the messages to the other neuron cell. So it's like just a brief introduction about the anatomy of a neuron cell. And neurons communicate with each other through a combination of electrical and also chemical messages. And the transmission of the signals actually here within the neuron is electrical at in the beginning. This video actually is not working very well here, but it's showing that in the beginning, the transmitting of the signal is like starting with electrical transmission, and then we have after that this chemical transmission between these two neuron cells, and then going to the other cell. And the combination of these two electrical and chemical signals are going to allow for transmission of the information between the neurons and between neuron cells, and enable different functions of nervous system, such as motor control, cognitive processes, and sensory processing. Another important thing that we are going to talk about this here is about the action potential, which is a rapid change in membrane potential, which is happening when the neuron receive a signal from other neuron cell. And the action potential is a rapid and transient change in the membrane potential of a neuron. which is really important for transmitting the electrical messages along the neuron cell. And it's actually a key mechanism in the communication between the neuron. We are going to actually using the differential equation to model this membrane potential behavior. So, and action potential actually is going to travel down from cell body through the axon and then end up these small branches. So through this process, actually, we have a change in the polarity, which is happening through the axon. We have here along the axon, we have different ion channels, like sodium channels, potassium channels, which are going to open and close. And they let this membrane potential actually going through the axon. And then we have generation of action potential, which is transforming the messages to the other neuron cell. And all of this opening, closing the channels here, which is going to cause this change of polarity, is going to transform messages in one direction to go to the other cell. And transforming actually neural impulses from one neuron to the other one, how there are different steps that should be done to transform these messages. In the beginning, we have sodium ion channels which are going to open. And these sodium ions are going to come from the outside, they're going to come inside of the cell. This stage of the action potential is going to call depolarization. And we have increasing the membrane potential. But it's not going to start until the neuron gets enough impulse from other, for example, neuron cell, any input current. If it is enough, then the neuron has the ability to spike. So for this depolarization, we need at least a threshold that the neuron get to that threshold. And after some point, when we get to a peak for the membrane potential, we have repolarization stage, which is in this number 3, this stage number 3, we have the sodium ion channels are going to close, and then we have opening of potassium ion channels. And then as you see, the membrane potential is going to decrease until to get back to the resting state of the neuron. So these are like the process that's happening through just one spike or action potential. And this generation of action potential actually is not only happening through the axon, but also the dendrite also. We can see that there are also showing this generation of action potential across all of these branches. So we can, this is something that is proved that. So we have across all of the parts of the neuron cell generating spikes and action potential. But how we can use a mathematical language to model this communication? So as we said, the neurons communicating with each other with generating this action potential. That is going to propagate through axon and to the other neuron cell. There are different models which is are available, different type of differential equation actually, we can say, to model this phenomenon, which maybe you have heard about the Hodgkin-Isley model, or for example, a very simple model of its Negumo or Maurice-Lécar model. Between all of them, we are going to talk about the last one, which is a simple model, but it's going to keep the biological properties of a Hodgkin-Isley model. That's why, so we select a Maurice-Lécar model. But what is this model? The Maurice-Lécar model actually describes neurons as a circuit with calcium and potassium voltage gated ion channels. And also we have a leak channels here. We can see the circuit in the right-hand side of the slide. And calcium and potassium channels are used because Maurice and Lécar, they found that the barnacle muscle fiber production of oscillatory behavior is dependent on external calcium concentration and potassium conductance. And in our body, actually, neurons are in a salty like a solution. So there are ion channels, in particular potassium and calcium ion channels, that generate electrical potential, which are moving in response to the potential. And when ion moves actually through the cell membrane, the currents are produced as a result. So that's why we can think of the neurons as a circuit, and we can see here. But let's look at the this model, which is a system of differential equations. So the Morris-Lecar model is, as you can see, is a mathematical model which is used to describe the dynamics of membrane potential. The first equation here is showing the how membrane potential is changing, evolving through the time. So it's going to be the rate of change of membrane potential over time. V is going to be the this V capital here is the membrane potential. The first equation we can see, and it evolves through the time. And the second equation is showing the evolution of gating variable. N is a gating variable parameter which represent the activation and inactivation dynamics of the ion channels. And it's going to control opening and closing of all of those ion channels across the axon, which is actually controlling the and changing the membrane potential. And they can be, for example, here we have potassium and calcium and leak ion channels. And there are other parameters, for example, here in the bottom of the slide, we can see that M infinity, N infinity, these are like representing the steady state values of the gating variables. The tau N is a time constant associated with the gating variable, which we can see the activation kinetics of a specific type of ion channels. So we use all of these parameters like different activation parameters, and I'm going through these more parameters in the next slide to model this generational action potential. So it's actually the simplest model to represent that. So these are the list of biological parameters for the Morris-Lecar model, and maybe we can talk about like them later on. But we are interested in applied current. Actually, we are going to change it to see what is going to happen to the dynamics or behavior of the neuron cell. And the C capital here is the membrane capacitance. And also we have the temperature factor, which is interesting. If we change, for example, what is going to happen when we change the temperature to the behavior of the neuron. So in a dynamical system actually is a part of a differential equation. We know that when we are changing the parameters of the model, a qualitative change actually happens in the dynamics of the model. And for example, these ones are going to appear or disappear, or we have other type of behavior. So it is changing behavior in the dynamical system point of view, or in the language of differential equation, is called bifurcation. So saddle node and half bifurcation are very common bifurcation in neuroscience, actually, that they describe the single spike properties of a spike generation mechanism of the neuron. And there are different, like many ionic mechanism of spike generation, but there are only 4 different types of spike bifurcation which are describing all of these different mechanisms. So one of these, as we said, is half bifurcation. So it's the transition from a stable steady state to the stable limit cycle when we are changing the parameter. And also we have another type of bifurcation which is called saddle node bifurcation. So through the half bifurcation, actually we have a stable focus which is going to lose this stability when we are changing the parameter, and a limit cycle is going to appear. We are going to see actually when we are simulating the solution of the Morris-Lecar model. For example, here we can see the for different applied current 60 and 100, we can see different behavior of this neuron cell, single neuron cell. So for the first and the left-hand side, we can see some of this solution or membrane potential of the cell, they have the ability to fire a spike, and then going back to the resting state, but some of them, they don't have this ability. So they didn't get to that threshold that they need to fire a spike. In the right-hand side, we can see with increasing the current, actually, we see the different type of dynamic or behavior. We have a periodic actually solution for this model. In the right-hand side, we can see that. Actually, in this slide, we can see that how is generation of half bifurcation, like in neuroscience point of view, actually, in the, for example, this one, the top one, when there is no applied current for the cell, there are some of these neurons that they have ability to fire a spike. They go back to the resting state, but some of them, they don't have this ability to a spike. when we increase the current, for example, very big value like 87 or 88 or 90, we can see a limit cycle is going to appear. And this is the point actually we have half bifurcation in the system. And also the interesting thing here, we can say that we can talk about the threshold. The point that at which, for example, we can see that some of the neurons or solution of the system or trajectory of the system, they have ability to spike, but some of them they just go back to the steady state. That's why we call this point as a threshold for the model. Also, if we continue more through the continuation of fixed one, we can see generation of this half bifurcation, and we can find out the value for the membrane potential and this gating variable n for that. Also, it can be interesting to find a bound for the membrane potential, maximum, minimum membrane potential for the having a spiking behavior for the neuron cell, which is again through the continuation of equilibrium point, which is happening here. But there are another type of modeling in differential equation, which we call them fractional order modeling. When we have integer order model, in those type of modeling, we just see one model and we cannot see different classes of the model. But when we have the fractional order, we can use a parameter like which is actually the that fractional order parameters to control the behavior of the model, and it's going to give us different classes or different dynamical behavior of the model. So it's another interesting way of modeling this communication between the neuron cell, which is called fractional order modeling. So we are going to explore different dynamical classes of the Maurice-Lecar model. So in here, we are using the language of actually fractional or like calculus, and we are using this fractional differential operator. We write down our equation for the first one and second one, the gating variable, using this fractional operator, and we define the fractional derivative. There are different types of for that. For example, we have capital, or we have, for example, Riemannian Liouville operator. We can use it, and we write down this fractional derivative in this form. And then the next step, actually, we need to discretize the model that we have. So we need to do some approximation. One of these methods for approximation is called like Graham-Wald technique of approximation. This actually is based on non-standard finite difference method. There's another actually numerical method within the theory of differential equation. We apply it to be able to discretize the equation that we have. And then we can find, after discretization, we can find this fractional order form of this Maurice-Lecar model. But we are going to see actually now in the next slide more interesting dynamical behavior for the Maurice-Lecar model. So when we had only integer order model, this dashed lines are showing only the integer order model, the first model that we saw in the beginning of presentation. But when we use this fractional order modeling, which is another type of differential equation modeling, we can see there are different type of behavior for the this one single neuron cell. So again, we have for the first series of this neuron cell, we can see some of them, they have ability to spike. Maybe they reach to that threshold, and we can see actually for the in the middle, we can see some periodic behavior for some of these time series here. The solution of the system that we have generation of this limit cycle, which is showing the creation of half bifurcation, and also there is another type of bifurcation which is called homoclinic bifurcation, which is through this bifurcation we have a periodic orbit at the exactly at the time that we have the bifurcation in this beautiful periodic orbit happening here. But when we are moving far from the this bifurcation point or control point of the system, then we don't see any periodic orbit anymore. And so this fractional order modeling was another way that we could look at different dynamics of for this single neural model. There are different actually approaches that also we can use to model, for example, dynamics of a brain through just using multi-scale modeling, for example, to show different types of dynamical behavior in the brain. So these are novel advances in mathematical, for example, approaches, and they are using different theories like topological methods or computational methods to extract different scales that we have. So we are talking about macro scale and micro scale. We need to model both of these type scales in the network of neurons that we have. So we have different actually topological or computational methods that we can use to display this multi-scale dynamics of the model that we have. So there are two main approaches to analyze modeling of the network, which you're going to see here, some of these models, but these are still working on that. And for example, one of the current study that is very interesting to do is like in multi-scale modeling of brain dynamic would be, for example, we have three main categories, which is the first one is we consider intrinsic properties of the cells within the neural networks. Another one, we are going to consider synaptic properties of the neural connections, or maybe we are going to consider topological properties of this network connection. The first and second actually they are containing this multi-time scale and depend on the parameters of the system. But the third one, the topological, which is based on the topological properties of the network, they don't include the multiple time scale, and they depend on the parameter of the network that we have. So we are now talking about the network of the neurons that they are communicating together to do or to perform a certain task. So the first one, when we talk about intrinsic properties of the neuron cell, we need to include the channel gating variables, which are very important to describe the neural cell dynamics. And these channels may be like activate or inactivate and this disparate time scale. And also we assume that we have a general two-dimensional neural model for the first part. And here V is actually membrane potential and W is the channel gating variable, which displays the channel state variable. And the second one was this, based on the synaptic properties of the neural connection, that we follow the following three-dimensional differential equation actually here to model a pair of coupled neurons. So again, we are talking about a network of neurons, but we are going to include the synaptic properties of the neuron connection. And so these are the parameters that we had before, then included this here. Again, the same model that we have. The third approach that for doing this multi-scale modeling again, which is based on the topological properties of the network, we assume actually that our network is like sparse or dense. So we have dense connectivity, and we assume that the cell communicate with a small or large number of other cells to perform the task that they do. And of course, there are actually the other type of network architecture, but what we can see here is just three different approaches that we can use for doing this multi-scale modeling of the network. So through this presentation, we became familiar with the different type of modeling in differential equation, like just integer order modeling or fraction order modeling, or how we can do this multi-scale modeling to be able to demonstrate how the neurons communicate with each other, how we can only, even modeling one single neuron is going to be very complicated. When we are talking about the network of neurons, we need to include different time scales, like macro scale and micro scales. So I'm gonna finish a presentation here. I think we have time. I don't know for any question. And thank you so much for your attention.
Yeah, I have one question, if you don't mind.
Yes. Yes.
Can you hear me okay?
Yes. Yes.
Okay. Yeah, I'm a mathematician, but one of the reasons I came here, and maybe you can shed some light, but one of the reasons I came here, with the neuron caught my eye because about 4 years ago, I had a real bad case of COVID, and it triggered neuropathy in my my peripheral neuropathy and some autonomic neuropathy, and I'm doing much better now. But I was just curious about that because I'm very smart on nerves now because I've studied Dr. Google, and so I've learned a lot about nerves. I'm just wondering if this is can this be applied to illnesses such as that?
So actually, it's a good question that you asked. We have, for example, for when we are looking at the interbrain and different brain disorders, then we are talking about the network of neurons.
Right.
What is happening, for example, between the neurons. Then the approach that we use, for example, the topological approach is we need to use graph theory-based method because we are going to look at the brain network as a graph, as a weighted graph. Then through the different topological methods in algebraic topology, we can characterize actually different behavior for this network and what happened. For example, we can actually characterize control a normal, healthy network and a network which any, for example, disorders that happen in the functionality of the network. So for the case that for different disorders that happening, like different, even in different other type of brain disorders like Alzheimer, I don't know, other type of brain disease,
Right. Yeah.
epilepsy, yeah. I think the graph theory based method would be perfect because these models are just for a single neuron model. But when we are talking about the all of this part of the different brain regions and how they communicate, which part affected through this and maybe malfunction of some neuron cells is something that we need to really use graph theory based method.
Okay. Thank you very much.
Thank you so much for the question. Any other question?
Oh, there's a hand up there.
Yes. Maybe in the chat.
Yeah, I see Kerry has his hand raised, so I was just telling you that.
Could you please, do you want to write in chat if you cannot? Maybe you can unmute. I was not able to do that. You can maybe write in, if you cannot use microphone, do you want to write in chat? Okay, yes. Okay, thank you so much. How do you approach a model testing for the, so for testing the model that we have, okay, for this single neural model, I didn't have data to validate my model actually. And but I worked with actually network of neurons, like I had like resting state data, and then to see, for example, if my results is significant or not. Definitely we have to perform different statistical tests, which is paired as a statistical test because we are gonna see that if our results are significant or not. So for this type single neural model, definitely having data, I mean, it was hard. I didn't have data really to validate the model that I had and check it with the, for example, this behavior that we got for example, this type behavior. But I had access to network of neurons data. Then it was easy. It was very easy to see if, for example, graph theory based models is going to be able, for example, to characterize different brain disease or not. But for this model, I didn't have actually data to actually. It's good because if we have data, we can check that which one of these, for example, model is going to be the best approximation for example, this single neuron cell. and we can actually support that. For example, which one of these orders for this fractional derivative is gonna better model this action potential, generation of action potential. But no, I didn't have data actually to test this or validate my model. But thank you for the quiz. It's a good comment. Thank you. Is there any other question, or should I? Okay. So I think if there is no question, so should I end the session now?